I'm Sharon Courtney, and we're talking to Dr. Tony Hu, the Weatherhead Presidential Chair at Tulane University, who's using applied engineering to develop a rapid test for COVID-19. How does your lab work with infectious diseases?
We're actually working on a very different format compared to the many other engineer groups. So in the field, there's a pretty the common practice, and the engineer team like to develop the highly sensitive platforms for biomarker profiling. But the lack of knowing better about the pathogen itself or the disease need or unmet need in the clinic, sometimes those the very novel sensors cannot be quickly translated to the application. So for us, and whenever we try to set up the research project, and we like to understand thoroughly what's the need in the clinic for this diagnosis.
Is there any particular case?
The particular case in this group is the detection of tuberculosis, and which is another very serious global health issues, and it's affecting millions of the tens of million people every year. And so, but the current gold standard can only cover 50% of them. And so we actually think out of the box, and it's trying to identify some new markers in the blood. And using this blood-based test, we can cover much more populations in the world, including the HIV-infected patients, pediatrics, also infant patients, and also the patients with extrapulmonary TB infections, which is generally the very difficult to be diagnosed using the current gold standard.
Why is testing for this virus so important?
Definitely, if the patient can be diagnosed earlier, and the treatment could be easier for the patient, for both patient and the doctors. And also the rapid test or the easy to operate test can also minimize the fear in society, right? And also with the better diagnosis result, and we will be able to know which patients should be isolated and minimize the spread of this virus.
How do the screening tests see or find a virus?
So right now, the U.S. heavily rely on the nucleic acid test, which is also not perfect. And based on the feedback from the clinic, we already see a lot of the false negative result. And so definitely there is urgent need for new technologies or the new biomarkers to address the current situation. And as I know in some other countries, and they combine the different strategy, including the nucleic acid test and also the CT scan, and some others. So this is quite different, right?
What is your lab doing to try to find a test?
Well, with better understanding about this pathogen and this virus and how they the functions or how they interact with the human body, and right now we're building up the multiple platforms, and for example, like the how to early detect those virus. So in the human bodily fluid, we also targeting on the nucleic acid biomarkers, and we're developing the CRISPR-based technology, which is hundreds of times more sensitive than the current kit provided by US CDC. And the second method, we can really convert the current platform for TB diagnosis to thoroughly work to comprehensively diagnose this coronavirus. And the third solution is so no matter you use nucleic acid or the antigens or antibodies, and the concentration of those biomarker is a concern. So if the concentration is quite low, and they will definitely trigger the issue in the clinic for false positive or false negative. So we're trying to identify some new biomarkers with greater diagnostic power.
Research often takes decades. How long will it take you to develop a test?
The CRISPR-based technology, and now we're working on it, and it could be ready for clinical validation anytime. The second one, we like to starting from the nozzle swab samples, and it really depends on the one the sample could be available for us, and we can directly start the technology development. And the third one also rely on the biomarker discovery, and that could be take at least 1 month to figure out the better solution.
This isn't the last time you'll hear from us, and we want to know what questions you have. So please email us at medquestions@tulane.edu.